Welcome back to Dr. Liu's VCE Biology. Today's topic is photosynthesis, cellular respiration, and biotechnological applications, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Yeast is used to help make bread. When yeast cells respire anaerobically, they produce a gas that helps make bread rise. Tha Darcy and Amal wanted to test how the different amounts of yeast used in dough impact the height of a loaf of bread. Each carried out their own experiment. They each used the same bread maker to bake loaves of bread with varying amounts of yeast. They then measured the height of each loaf of bread. All other variables were kept the same. All the baking was completed over the course of one day. The objective of this study is to quantify the effect of varying concentrations of yeast on the final loaf volume, where the yeast concentration serves as the independent variable, and the bread height serves as the dependent variable. Across trials, quantifiable data demonstrated that reducing the yeast concentration from 1.0 to 0.5 teaspoons consistently resulted in a diminution of final loaf height. This observed dose-response relationship was determined to be highly repeatable across all replicates. The question specifically asks for a factor that immediately influences the ATP yield during the electron transport chain, etc. Option A, incorrect. Glucose is processed in the cytosol, not the cristae, far upstream of the etc. Furthermore, the etc. does not directly use glucose. Option B, correct. Oxygen is the final electron acceptor in the etc. If O2 is not available, the entire chain backs up immediately, the proton gradient collapses, and ATP synthesis stops. This is the most immediate and critical limiting factor. Option C, incorrect. Pyruvate is converted to acetyl-CoA in the matrix, which feeds the Krebs cycle, which then supplies NADH and FADH2 to the etc. A change in pyruvate concentration is an upstream factor, not an immediate influence on the chain itself. Option D, incorrect. Glycolysis is a process upstream of the etc. While a faster rate eventually supplies more NADH and pyruvate, this influence is delayed and not an immediate factor acting within the etc. Option A, correct. Anaerobic pathways bypass the electron transport chain, which is the major source of ATP, oxidative phosphorylation. Option B, incorrect. Anaerobic respiration consumes the small amount of NADH it makes to regenerate NAD+, preventing it from entering the high-yield electron transport chain. Option C, incorrect. The Krebs cycle is not used in anaerobic respiration, as it requires oxygen to function. Option D, incorrect. Aerobic respiration involves the complete breakdown of glucose, yielding the highest amount of ATP, 30 to 32 molecules. An increase in some molecules most likely causes a decrease in the rate of glycolysis within a healthy muscle cell. Option A, incorrect. An increase in glucose would generally increase the rate of glycolysis, as it provides more starting material. Option B, incorrect. NAD+ is required for glycolysis to proceed, it's reduced to NADH. An increase in NAD+ would generally increase or sustain the rate of glycolysis, especially under anaerobic conditions where NAD+ can become limiting. Option C, incorrect. An increase in oxygen allows the cell to switch to the more efficient aerobic respiration, the Krebs cycle, an electron transport chain, which produces a large amount of ATP. The resulting high ATP levels then inhibit glycolysis. Therefore, oxygen causes an indirect decrease via ATP, but the immediate, direct cause of the inhibition is the increase in ATP itself, feedback inhibition. Option D, correct. Glycolysis is tightly regulated by the energy state of the cell. The primary mechanism for slowing the rate of glycolysis is feedback inhibition by its end products, particularly ATP. The light-dependent reactions occur in the thylakoid membranes within the grana of chloroplasts. This process involves the splitting of water using light energy, which releases oxygen gas, electrons, and protons. The electrons and protons are then used to generate the energy storage molecules ATP and the reducing agent NADPH. Light intensity is a primary external limiting factor for the light-dependent reaction because the process requires light energy to split water. Conversely, carbon dioxide concentration is the limiting factor for the subsequent light-independent reaction, Calvin cycle. Although ATP is a product of the light-dependent reaction and a key reactant for the Calvin cycle, its concentration is not a direct limiting factor for water splitting or NADPH production. However, the buildup of ATP and NADPH can indirectly limit the light-dependent reaction via feedback inhibition when the Calvin cycle runs slowly. Water serves as the electron donor and proton source in the light-dependent stage of photosynthesis. Option B, incorrect. The light-independent reactions Calvin cycle use ATP, but they do not produce it. Furthermore, they do not produce ATP from CO2 and H2O. Option C, incorrect. The light-independent reactions use ATP to produce glucose, but the light-independent reactions certainly do not produce ATP from glucose. Glucose is used to produce ATP in cellular respiration, glycolysis, which is a different process entirely. Option D, incorrect. The light-independent reactions convert CO2 and ATP into sugar, not the other way around. The light-dependent reactions do not form glucose. Option A, correct. The ATP and NADPH produced in the light-dependent stage are used to fix carbon, turn inorganic CO2 into organic sugar molecules. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.